Our laboratory studied T lymphocytes, also called T cells. They are a type of white blood cell and form part of our immune system. They help protect the body from infection by killing cancer or infected cells. In our experiments, we use T cells that are found in the lymph nodes, spleen, and blood. We keep them in plastic plates and flasks filled with liquid media that contains all the nutrients they need. These T cells are called naive. They are small and not very active. However, during an infection, they are activated and convert into effector T cells. For some of our experiments, we convert these naive T cells into effector T cells. This lets us study how they act during an infection. To do so, we activate naive T cells using an activator that we add to the plate. We place them in an incubator set at 37 degrees Celsius and 5% carbon dioxide and leave them for 48 hours. During this time, the T cells grow in size and rapidly increase in number. After this time, the liquid media has turned yellow, meaning that the cells have used all the nutrients present in it. We collect the cells by spinning them in a centrifuge and put them in a flask with fresh media to provide more nutrients for their rapid growth and increase in number. We add a cytokine called interleukin-2, IL-2. This helps T cells to convert into effector T cells and increase in number further. We leave them in the incubator overnight. In the morning, the cells have grown and divided, making millions of these killing cells. Now we count them. We prepare a sample for counting by adding 100 microliters of our liquid media to a tube. We go to the bench and add 900 microliters of PBS containing DAPI to the T cells. DAPI is a fluorescent DNA stain that only stains dead cells. We count the T cells on a machine called a flow cytometer. It can count up to 10,000 cells per second. We grow or expand these cells for a further 5 days, keeping them at a density of 0.3 to 0.5 million cells per milliliter. As they keep increasing in number, we need to do daily counts and replace the liquid media containing IL-2. The cells are stored in an incubator during this time. Now we know how many T cells we have, we can use them in all sorts of exciting experiments. We can study how they respond to different infections and how nutrients and their environment affect their ability to fight infection. One thing we can look at using the flow cytometer are differences in the features of the T cell. We can compare the size of naive and effector T cells. Effector cells are much bigger in size compared to naive cells. They are also more granular. This is because they have specialized granules containing granzymes used to kill the target cells. The data from the flow cytometer is shown as scatter plots. Forward scatter, FSC, is used to measure cell size, whereas side scatter, SSC, indicates the granularity. Once we have finished our experiments, we throw away our cells into liquid waste. Virkon, a biocide used to destroy biological material, is added.